Good morning, students. We'll start for the today's day. We are entering day 11 of our BIIS-11 workshop. Today we will see about the phytochemicals, extraction techniques, and analytical tools that are used in phytochemical extraction and to identify compounds. Okay. So is my slides visible? Everyone?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Thank you. So I will be giving the talk. So myself, Dr. Amarchand Kadiyamurkan, working as a scientist as well as a BIIS coordinator. Today's topic is phytochemical extraction methods. So generally, there are a lot of medicinal plants related research, ethnobotany research, ethnopharmaceutical research is going on around. So for experimental purposes or for purification of the compounds, everything is the major target in this research. So normally these phytochemical extraction involves extraction and also the determination of quality and quantity of the bioactive consume. So whatever compounds you are going to extract, you are wishing to extract, both the quality of the compound and the quantity, the volume of the compound you need for your experimental purpose determines the full process of this phytochemical extraction. So the primary method is like preparation and screening only. So screening, although like different methods are involved, lot of methods are involved in the extraction process. Choosing the correct method determines your next step in your experimental process. So if you are not choosing the correct method to extract a particular compound, molecule, biomolecule, then your further research studies, experiments will be affected by that. So always be critical while designing your research proposal. Choose methods which are more suitable for the compounds you are wishing to isolate and study. Okay. So before going into the details of the extraction procedure, we will learn some terms, basic terms that are used in phytochemical extractions. First one is medicinal plant. It refers to the plant containing an active ingredient or a secondary metabolite that possesses biological activity. Okay. So coming to the next one is herbal medicine. Herbal medicine means it is the preparation of a bioactive compound, bioactive ingredients. Okay. So there is a difference between a medicinal plant. When you call a medicinal plant, means it contains certain ingredient that possesses a biological activity. But herbal medicine means that particular biological property, the preparation which has the biological property is called herbal medicine. And one is next one is menstruum. Menstruum is the liquid or the solvent that we choose for the extraction process. Next, mark. Mark means the insoluble or inert material that is left behind after the extraction. So normally we extract with the different type of solvents, and the residue, the plant material or whatever is left behind is called mark. And mishal is the solvent that is used for extraction of the along with the extract. Next one is fingerprinting in medicine. So fingerprinting means there are different techniques, chromatographic techniques, different identification techniques, and chemical analysis that are used to categorize and characterize the bioactive compound is called fingerprinting of medicinal plants. And immunoassay. Immunoassays normally if you're looking for an like a proteins or enzymes that has an active as an active molecule, then we use different methods to identify that biological activity, like ELISA methods or receptor binding methods or immune reactions method, IgI, antibodies, using antibodies. Okay, these are called immunoassays. So next one is solvents that are used in extraction. So there are different types of solvents ranging from polar to non-polar is there. So the choice of the plant material for which part you want to extract of the plant, the type of plant material, or the nature of the bioactive compounds, and the availability of the solvent. These are the major factors. Okay. So then the two types of different types of solvent is there, polar and non-polar. Polar like water, methanol, ethanol, and non-polar is the other end like hexane, petroleum ether, and dichloromethane. So here you can find the different polarity of different solvents that are generally used in the uh, phytochemical extractions. So again, in solvent extraction, there is another factor called liquid-liquid extraction, in which two different immiscible solvents are used for separation of the compounds. And the compounds to be extracted using this should be soluble in organic solvent, but not in water, because we here in the two immiscible solvents, we use one as a water base and one as a solvent, any one organic solvent. And during the fractionation, solvents of different polarity, like base from hexane end hexane from low polarity to high polarity is normal. In this order is followed. So let's see some of the properties of different solvents that are used in the extraction process. So water is, as everyone knows, is the most polar solvent used in extraction of polar compounds, and which dissolves a broad range of compounds. It is very cheap, affordable, non-toxic, non-inflammable, but it may produce, water extract may produce, promote bacterial and mold growth, like fungi or yeast growth can be seen, which may degrade your compound. Okay. So and also may require heat or freeze drying method to concentrate your extract. And the next popular solvent used is alcohol. So either it is be methanol or ethanol. It is a polar compound and miscible with water. It is also used to extract polar secondary metabolites. So when you are using alcohol, like the growth of microorganisms can be prevented to up to 20 to 30%, because alcohols are normally as a disinfectant we are using, but it is also used as it will preserve your compounds from getting hydrolyzed by bacteria and fungi. And generally, this is a low toxic at low concentration. When you are using alcohol at lower concentration, it is low non-toxic. At higher concentration, it is toxic to cells. And only you need small amount of heat for concentrating the sample. So after you extract using large volume of solvents, generally you reduce the volume for your experimental purposes. You concentrate them. So only little amount of heat is required for alcohol-based solvents. The next one is chloroform. Chloroform is a non-polar one. It's majorly used for extracting terpenoids, flavonoids, fats, and oils. So soluble in alcohol, the compounds which are extracted from the chloroform are soluble in alcohols and absorbed and metabolized by body. Even though it has a sedative and carcinogenic property, the very low use of chloroform may be giving you a better result. So next one is ether. So ether is also a non-polar compound. It is used for extracting alkaloids, terpenoids, coumarins, and fatty acids. It is miscible with water, has a very low boiling point, and it is also very stable, that it does not react with any acids, base, or metals that you are going to use in your further experimental process or extraction process. So it is a highly volatile and flammable solvent. And the next is ionic liquid, which is also called as green solvent. It is highly polar and extremely heat stable. Even up to 3000 degree centigrade heat, it will remain as a liquid and extremely miscible with water and used for polar extraction. This ionic liquid solvent is used mostly in microwave assisted extraction, and it is non-flammable. So factors. So for selecting a solvent because solvent is the main important thing for extraction. So before you are going to confirm or use as solvent, there are certain factors that you need to check. That is selectivity. Selectivity means what type of compounds you are going to extract. You are either a polar compound, you are non-polar compound, you are fatty acids like the proteins or enzymes, whatever you are wishing to isolate. According to that, you should select your solvents. Okay. Then safety. Safety means it should be less toxic and then non-inflammable. Okay. And then cost. So some solvents may be cost higher, some solvents may be cheaper. So affordability is also a major point while choosing your solvent system. And then reactivity. Your solvent should not react with any metals that you are going. In case you are going to use some metal related apparatus or something, then it should not react with any metals. Okay, so the reactivity of your solvent is also very important. And then recovery. Recovery means in most of the extraction process, you can recover the solvent system. Some solvents are able to be recovered using different methods. Okay, so recovery means you can reuse the same solvent for the next extraction or other extraction process also. And viscosity. Viscosity, if higher the viscosity, it will not penetrate the cells of the plant tissues. Okay, so the viscosity should be lower so that it can easily break through the cell systems, and it will help in extraction of the secondary metabolites you are planning for. The last one is boiling temperature. So boiling point of your solvent is also very important. In certain methods, we are going to boil the solvents to extract the compounds. Okay, so lower the boiling point, the better the compounds will be. You can get a good quality and quantity of compounds, because the quality is main important thing in all phytochemical extraction. Major important thing in phytochemical extractions. If the boiling point is very higher for a solvent, then your compound should also be heat stable. So if it is not thermostable, if your compound is not stable at a higher temperature, then it will break down and you will not get the compound which you are planning to extract. Okay, so boiling point is one of the major factors. Next we see different extraction methods. So maceration. So maceration is generally like a coarse powder. You prepare the dry materials and then dry the plant material and prepare a coarse powder and mix with the solvent in the ratio of 1:5. Okay, and then you put it in a container or a glass beaker, and then you allow it to stand for 3 days with occasional shaking. And then after the 3 days, your mixture is separated from the mark by normal filtration or decantation technique. The solvent is concentrated by evaporating in using a oven, water oven, or a water bath. And this method is best for thermoliable drugs. All the compounds which are breaking down in the heat method, you can go for this method. This is the simplest method, and simplest and best method which can use for most of the drug compounds. So second method is next method is infusion. Okay, so in infusion it is similar to maceration, where we use fine powdered plant material inside a container, and hot or cold solvent is poured on top of the powdered material, and so for a very short period. Not like in maceration we keep it for 3 to 4 days, but here it is very short period only you will do. And the best example, like every day we used to do the green tea preparations, right? So similar type of preparations is called infusion, and it is suitable for bioactive molecules that are readily soluble. So we are going to only expose the plant material in a very short time. So in that short time, what are the molecules that are readily dissolvable in the solvent will come out. Okay. So this is for in case of you want the fresh extract. Always need a fresh extract for your experiment, you can go for this method. And normally the solvent ratio is 4:1 or 16:2. 1. And the next method is digestion. So in this process, a moderate amount of heat is used. The solvent and the plant material is kept in a container in a water bath or oven at 50 degree centigrade. The heat is applied, constant continuous heat is applied, so that whenever you apply a heat to a solvent, the viscosity of the solvent changes a little bit. So it helps in and enhances the extraction of the secondary metabolite. And it is also suitable for the readily soluble compounds. Coming to the next method is percolation. So most of you have seen a similar type of apparatus in your homes where people have a habit of taking coffee, like filter coffee. So same type of unit is used here. The apparatus is called a percolator. Fine dried plant material is moistened with a solvent of extraction and kept for 4 hours. And after 4 hours, the content is allowed to stand for 24 hours. And then the lower part is opened and then the liquid is collected. Okay. So this is also the extracted collected liquid is then filtered to get a pure compound. The next popular method is Soxhlet extraction, also known as a continuous hot extraction method. And the apparatus used here you see is called Soxhlet extractor. Okay. Fine powdered plant material is placed inside the thimble in a clean cloth or wrapped in a clean cloth or a filter paper. And the extracting solvent is heated from the heating mantle situated below from the round bottom flask. So the solvent evaporates and passes through the condenser, and then it condenses down and it comes in contact with your solvent. So okay, once contact with your plant material. So once this process is repeated several times, following the until all the extract is extracted. Okay, this is suitable for a material for partially soluble and thermoliable. Okay, so if your compound is not stable to heat, you cannot follow this method because we are using continuously heating the solvent and then using. Okay, so large amount of drug compounds can be extracted with small amount of solvent system, and no filtration is required, unlike the previous methods we saw. And the next one is microwave assisted extraction. This is one of the advanced extraction procedures, and we use the dipole rotation and ionic transfer by displacement of charged ions present in the solvent and drug material. So here we apply a radiation of around 300 megahertz to 300 gigahertz of energy is used, mostly suitable for polar solvents. Minimizes the use of solvents and extra in short time period. You can use because you are applying a large amount of electromagnetic radiations to break down the cells and to get the compounds out. Very small time is required for the extraction. And most commonly, this method is used for extraction of phenolic and flavonoids. You can see the setup where your round bottom flask is kept inside with the solvent, and the compound is kept inside the microwave chamber, and it is adapted. And the outside of the chamber, there is a tube that runs coolant condenser tube, which condenses the solvent and it is collected. Extraction is collected. And the next method is ultrasound assisted extraction method. Here we use ultrasound frequencies from the range of 20 kilohertz to 2000 kilohertz. This ultrasound breaks the cell wall, plant cell wall, and increases the solvent penetration for the release of secondary metabolites. Applied for mostly used for small quantity of samples, minimize the usage of large amount of solvents, and maximizes the yield. Okay. The method is difficult to be reproduced because high frequency, because we are using 20 kilohertz to 2000 kilohertz energy. So these energy may sometime degrade your phytochemical compound by producing free radicals. Okay. So this method is not reproducible. Okay. And one more method is supercritical fluid extraction. This is also called as a green extraction technique, where we use carbon dioxide as a supercritical fluid, and sometimes along with carbon dioxide gas, we use co-solvents such as methanol or ethanol. So these using of co-solvents improves the selectivity, higher yield extraction, and better fractionation capabilities. So applicable to all hydrophobic compounds, those compounds which cannot be extracted in or hydrophobic in nature can be extracted in high efficiency by infusing the polarity of supercritical carbon dioxide. Okay. So polar compounds can also be extracted by addition of polar modifiers that like ethanol and methanol. So similar to the methods methods of extraction, there are certain factors, important factors, which we need to consider before choosing a method. Okay, so the method we are choosing to extract should be, the compound should be stability to heat. Okay, so too much of heat should not be applied depending upon if your compound is like a thermo liable, then you cannot go for a higher heat method like Soxlet, yeah, ultrasound, yeah, super microwave assistant method. You cannot. Okay. And then nature of solvent. So mostly the nature of solvent should be non-toxic and non-inflammable. Okay. So if it is toxic and inflammable, then the extraction method you need to choose a different solvent carefully. And then cost of drug. So if normally if you are preparing a crude drug, you can simply go for maceration technique. Okay. Where less amount of solvent is used. But in case of you want a purified compound, means you need to use lot of solvent system to separate the molecule, to isolate a single compound. Okay, so the cost of drug, the molecule which you are planning to use, also determines the extraction method. Okay, and then duration of extraction. So short time duration is also some compounds is used to extracted using in a very short time, like ultrasonic sound you can use. Okay, that will like 30 minutes you can get a compound. Okay, fraction extract. But in maceration you have to wait for 4 days. So depending upon the duration which you want to use, depending upon the compound you are planning to extract, the duration of your extraction method is also a major factor. And then finally volume required. So in like we saw in ultrasonic assistant, supercritical fluid as extraction methods, the extraction compound volume, final volume of the extract will be very less. Okay? So, but in case of Soxlet, maceration technique, percolation technique, the volume of the extract you are going to get is a good amount of volume you will get. So that you can also get a more amount of compounds that you are planning to. And then intended use. Okay, for what purpose you are using, what kind of purpose application your compound, your extract you are going to use. Okay, if you are going to use only like a topical application, you need only the crude extract, or you need either you need a purified compound as a drug compound to using a drug delivery system. You are going to take the molecule and prepare a drug. Okay, so based on different types of the applications you are planning, you can choose your extraction methods. Next, we see fractionation and purification methods. So fractionation is a process of separation of plant extracts into various fractions, which is further separated into different fractions of different fraction until you get a pure compound. Okay? So two method is used, basically a chemical method and a physical method. So for the first method is separation funnel method. Okay. So different solvent system here is used, like N-hexane, chloroform acetone, ethyl acetate, N-butanol are selected. And the fractionation of crude extract is done by complete dissolution in water. Okay. So you dissolve your crude extract in water, and then you use different polarity of solvents to extract the different compounds. Okay. What are the compounds that are dissolved, able to dissolve in different solutions? That will go separated into that solvent system during this method. So here we see the normal separating funnel here. Okay, the separating funnel is used, and then you have two layers like organic layer and an aqueous layer. Okay, organic layer is your solvent, and aqueous layer is your extract dissolved in water. So you mix these both the layers vigorously and then allow them to stand for a few minutes to a few hours, and then both the layers get separated. Okay, then because of the higher density, the liquid the liquid which has higher density settles down, and first it is collected carefully, and then the organic layer is collected carefully and then concentrated. So this process is repeated like 3 to 4 times or 2 to 3 times or 4 to 5 times, depending upon which other solvents also. Okay, same solvents also we can use, and we can also use like different solvents. Like first time you do with N-hexane, next you can perform with chloroform, next you can perform with ethyl acetate or acetone, and finally you can do with N-butane. So you can use a different type of solvents in for one extraction method, and you get different fraction mentions, like you N-hexane fraction, chloroform fraction, acetone fraction. Each fraction you can get different type of fractions, and each fractions can be tested, concentrated, and then tested for biological activity. So the remaining portion that is left after the extraction is called residual aqueous fraction, RAF. The next method is fractional distillation. Okay. So the process of separating or purifying compounds of a mixture is based on the difference in their boiling point. Okay, so the compounds evaporates with the solvent, and each compound is fractionated. Compound fractionated will condense and collected separately through several siphons. There are siphons from the condenser attached to the condenser. So here we see that instrument. It is called a distillator, fractional distillator. So this instrument, you can see the water bath. Water bath is there with the flask attached, containing your extracted solvents. You can set the temperature of the water bath, and you can according to the solvent boiling point. Okay, so when the solvent reaches the particular boiling point, the vapors goes through the condenser, and then it condensates and collects in the siphon attached to the condenser. And the next method is called fractional crystallization. Here, large number of compounds that exist in nature are crystalline state. Separation is achieved via formation of crystals from concentrated extract using heat or refrigeration. Okay, here in this picture you can see whey protein concentrate is there. It is dissolved in ethanol, extracted in ethanol, and boiled, heating to boil and filter and evaporate this solvent. Okay. So when you boil this one, you can get the lipid extract. You are trying to separate the lipid extract using ethanol fractional distillation. So like this, each particular compound can be extracted. The next method is sublimation. This ancient method. The method used involves changing the compound from solid to gaseous state without passing to liquid state. Okay, the substances, the volatile substances like oil and camphor can be purified using this method. And coming to the chromatographic techniques. Okay. There are different chromatographic techniques that are used to separate the pure compound from the mixture of extracts that you have prepared. Okay. The separation is based on size, shape, and charge. So it also involves using of one mobile phase and one stationary phase. So the stationary phase is mainly silica gel, sapphadax, saccharose, aluminum, cellulose powder, and silane. And mechanism of separation is either through adsorption, partition, affinity, ion exchange, or size exclusion. And then the different techniques that we are going to follow is paper chromatography, thin layer chromatography, column chromatography, gas chromatography, and liquid chromatography. So coming to the absorption chromatography. The separation is based on interaction between the compounds and the stationary phase. Hydrophobic and non-covalent van der Waals forces of attraction are involved. And then loosely bound compounds are eliminated first. So coming to the picture, here you can see the column is filled with the matrix made up of either silica or aluminum gel, and it is completely saturated with the solvent system. And then you add the mixture containing your extract, that is your crude extract, and then you add a particular solvent. And then at the bottom, you collect the fractions. Okay, so the compound which has more affinity towards the silica or alumina will bind to the column, the silica material beads, and then it will take some time to elute. The compound which does not bind to the silica or aluminum, that means that shows a very weak van der Waals force. It elutes first. Okay, so in this picture you can see the Y compound, yellow color compound. It comes more quickly than the X compound. Okay, because the X has more affinity towards the matrix, so it needs more time to elute. So like this adsorption, simple adsorption technique, you can purify your compound. The next one is partition chromatography. So in partitioning chromatography, bioactive compounds are separated by addition of 2 or more immiscible solvents. The solubility of the compounds in a particular solvent is the major determining factor for choosing that solvent. And again, 2 immiscible solvents are used here to separate the compounds, and this is also known as liquid-liquid partitioning or solvent-solvent partitioning system. Okay, in a separating funnel which has a 2 molecules line. Color here, shown here, and then another solvent is added, immiscible solvent, and both are mixed, and then you can see the separation of the molecules in the 2 different layers. 2 different molecules get separated, and then after a short period of time allowing them to stand, you collect both different solvent systems separately, and each solvent system will have different compounds that we can further purify. Next one is affinity chromatography. So affinity chromatography, the stationary phase. In normal chromatography, you use a silica method, silica-based stationary phase. Here you use ligand-based. Okay, the stationary phase is a ligand which is positioned in the separating column. As you see, a mixture of protein is added to a column, polymer column, which has a ligand bind of a particular protein, interest of protein. Okay, so when you add the mixture of protein and then you add a solvent system to elute, the protein of interest will bind to the ligand. Okay, so whatever the ligand you designed for the protein of interest, the proteins will bind to the ligand, and all the other materials, unwanted proteins, will be first washed through the column and collected and kept separately. Then after to remove the protein of interest, to get the protein of isolate the protein of interest, you add a solution of ligands, more concentrated solution, and thereby the protein of interest is eluted, okay, in a solution and so these kind of techniques is used for mostly for isolation of proteins and enzymes if you want to purify. Okay, and then coming to the next one is ion exchange chromatography. So in ion exchange chromatography, the separation is based on the ionizing charge, ionizable molecules based on the total charges like proteins. The molecules which have ionization charges are proteins, peptides, amino acids, and nucleotides. So you can purify these type of molecules using the ion exchange chromatography technique. So the similar type of molecules are separated readily by changing the pH, buffer pH. Depending on the pH of the environment, proteins may carry a charge is either a negative charge or a protein positive charge, or a even no charge. Neutral molecules also can be separated using ion exchange. The pH at which the molecule has no net charge is called isoelectric. Okay. So here you can see the polymer which is having negatively charged functional group is packed in the column, and then a protein mixture is added. Okay, and then the negative molecules, because this is already a negatively charged functional group, so it will always attract the positive charged molecules, and the molecules which have large net negative charge are first eluted. So likewise, when you change the buffer, if you want to collect the positive charged molecules which are bind to the negatively charged polymer beads, then you need to change the pH of the buffer. So by changing the pH of the buffer from a different pH, you will be able to isolate different protein mixtures containing different charges. Okay, either a positive charge or negative charge. The next one is size exclusion chromatography. No name itself. So the name itself tells you it is based on the size of the molecular size. The separation is based on the molecular size. And it is also popularly called as gel filtration technique or molecular sieving technique. Okay, so here you can see the column is packed with the poorest material, and then the sample is injected with the flow of solvent. Okay, so size, on the basis of size, large molecules normally get separated first, because large molecules will never bind to the porous packing, and it will be able to elute through the through the column. Okay, so the first elution large molecule, it first comes down, and then the smaller molecules which are bound to the packed material, column material, will be eluted in the last. Okay. Okay. So next is chromatography techniques. Okay. So coming to the chromatography technique, the first one is paper chromatography. Okay. So paper chromatography is based, the mechanism of separation is adsorption. You need only a very simple apparatus, glass chamber and a filter paper made up of cellulose for different separating the identification of a compound. Okay. The filter paper is loaded with the sample and it is suspended in a glass chamber containing a mobile phase. And the mobile phase will be the solvent, different solvent systems. Either a single solvent or a combination of two or more solvent system can be used to separate the compounds for identification. And the mobile phase ascends the filter paper. Once you introduce the filter paper into the solvent system, the mobile phase ascends the filter paper via capillary action method. And the compounds that are readily soluble in the solvent system will be the first to get separated and moved. The RF value of each compound, each spot is calculated, and it is able to identify the compound. It is a very simple and cost effective and also very sensitive method. Only disadvantage is fragility of the paper. Okay? So you cannot reuse the papers in this case. And the next method is thin layer chromatography. Here also the mechanism is adsorption mechanism. Okay? So the separation is based on the interaction of compound between the stationary phase. In paper chromatography, we see the separation of interaction based on the interaction between the compound and the mobile phase. Here it is the interaction of compound between the stationary phase and the compounds. Okay? Ideal for low molecular weight, separation of low molecular weight compounds. And the stationary phase mostly is silica gel, powder coated silica gel is used. Similar to the same thin layer chromatography, paper chromatography, here also we calculate the RF value. And in this case, we can also compare with known standards to identify the molecules. The spots that are separated in the silica gel can be scrapped and redissolved, and further can be purified using different solvent systems. If you are unable to separate a very closely bound molecule, two molecules bound very closely, you want to separate, then you can scrap that particular region and you try with the different solvent systems for separating both the compounds. That can be done using thin layer chromatography. This is a less time consuming than paper chromatography. The spots are very clear and the compounds are very stable in acidic mobile phase. And the next one is column chromatography. So this involves several mechanisms like adsorption, molecular sieve, or ion exchange methods. Okay, mechanism. The column is generally made up of a glass tube, and the material stationary phase is usually either a silica gel, Sepharox, or Sepharox. There are different types of molecules, okay, that are used. It has a different bead size. Okay, the bead size determines the porosity of the column material, stationary phase material, which will also helps in determining the elution of your compounds. Okay. From polar to non-polar solvents, different mobile phase can be used in this method. Okay. Fraction of solvents collected individually at specific intervals, and then finally the pure compounds are pure. So here we see the stationary material is packed in the column, and then the mixture of to be separated is added, and the mobile phase is added. Okay, you can see different colored compounds, that is different compounds with the different molecular weight or adsorption based on the affinity towards the stationary phase is eluted at different time intervals. So each fraction of like a different volume, you can at different time you can fix a time interval like like 10 minutes or 15 minutes, or you can based upon the volume like 10 ml fractions like that you can collect, and then you can analyze the compounds using spectroscopy and then pool all the compounds similar that compounds that fractions which contains similar wavelength absorption wavelength in your spectroscopy. You can all pool them into one fraction, and then you can identify go for concentration and identification of your compound. And the next one is gas chromatography. So gas chromatography is a very popular and advanced technique used for partitioning of compounds using two immiscible solvents. One is a gaseous phase and another one is inert solid stationary phase. The substances that are soluble in gaseous phase will leave the liquid and get separated. Inert gases such as helium is used as at a constant flow rate. So in all the other systems we saw liquid system, liquid solvent system to use. Here we are using gas as a mobile phase. Okay? So crude extract is dissolved in solvent and is injected into the system. Here you can see in this figure, there is a sample injection port is there, which is connected to a carrier gas flow controller and a gas cylinder. Okay? That gas cylinder is mostly a inert gas. Okay? And then the sample injector is connected to a column. Okay, the column, spiral column, it will be like 5 meters, minimum of 5 meters of length. Such a long column, spirally wound column, is placed inside a oven, water oven. Okay, and it is maintained at a high temperature. Okay, so when the sample is injected and the gas flow is started, the normally and it passes through the column. So whatever the sample that is able to, once it reaches the column oven temperature, due to the high oven temperature, the compound present in the solvent system is evaporated and it gets mixed with the gas. Okay, that is flowing through. Okay, and then the gas will carry the molecule throughout the column. So here also affinity plays a major role. If your compound is attached, has higher affinity towards the column, it may stick to the column and it will get eluted later. Those compounds which do not have affinity towards the column will get first eluted and the detector will detect, and you will get the electrical signal is recorded, and the data processing is software helps you in identification of the compound and retention time. Based on the retention time, the compound is identified. And then HPLC. Okay, high performance liquid chromatography. These are mostly the compounds that are dissolved in solvents, that are able to dissolve in different organic solvents, or analyzed using HPLC system. This system allows both qualitative as well as quantitative analysis of your compounds. You not only can determine the purity level of your compound, you can also quantify how much micrograms or milligram levels of compounds are present in your extract injection. Okay, single injection. Okay, the solvent system is used for separate the compounds. Here we have a pump, solvent delivery pump, which will create a force. Okay, it will create a pressure inside the column while flowing through the column, and the sample is injected in a injector. So when the sample reaches the stationary phase, the column, which is again a material like silica gel or aluminum or Separax or Sepharos column is there. The affinity of the molecule towards the column determines the, and the solubility of the compound in the solvent system determines the separation and purification of your compounds. Okay, so the low affinity molecules and the molecules that are readily soluble in the solvent system are first eluted and detected in the system. Okay, so in the system. Likewise, if you want to gradient system, some molecules cannot be used in single solvent system. In that case, you can use more than 2 solvent system, or a gradient solvent system can be used. And the solvents are normally acidified a little using a trifluoroacetic acid or trichloroacetic acid can be used to acidify the compounds solvent system, so that the elution rate will be better, and the peak values, individual compounds detecting level will be very good. Okay. Okay. So these are the different basic extraction and fractionation procedures that are followed in for the experimental purposes in preparation of medicinal plant extracts and purification of compounds. So now, thank you for all you for listening. The session is open for discussion. Any questions? Any questions? Okay, Hexika. I will share the PPT with you. I'll share it in the group. Any other doubts or questions? So if no questions, we will move on to the next session. So Dr. Abhijit Kate of NIPER. Sir, okay, sir, in phytochemical extraction, we have to do characterization, but cost is high. Can we use alternative methods? Characterization in the sense like what? What type of characterization? Characterization for identification of the molecule. Nikshita?
Yes, sir.
Okay. So,
Sir.
so normally if you want to do like, there are different phytochemical methods are also there, like for identification of phenols, flavonoids, saponins, like this different class of molecules. You have certain biochemical test is there. That is the simplest method to identify the different biochemicals present in our crude sample. But in case if you want to identify pure compound,
Sir,
then
for
yes,
SEM, TEM, and FTIR.
SEM-TEM is not mostly used for the surface analysis. Okay? So for identification of the FTIR gives you the functional group present in your molecule. Okay? So if your molecule has a lot of functional groups, it can be analyzed using FTIR spectrum. For the structural analysis, you need to go for NMR technique or LCMS or GCMS technique that will give you the full structure and molecular weight of the compound.
Okay, sir. Thank you, sir.
Any other questions? Yeah, I will share the PPT. So Dr. Abhijit Kate, he may not be able to join us. So we will see his previous lecture. I will play for you. You can go through. He explains the various instrumentations, analytical instrumentations that are used for identification of a phytochemical or pure compound. So we will also see this. And then if you have any questions related to, you can write to me. I will try to get answers from him. Okay. So how can we identify the phytochemicals from HPLC retention time and peaks in absence of standards? Okay, good question. So in the absence of standards, you have no other choice but to compare with other established methods. Okay. So normally we do the methods, we optimize the conditions for HPLC, and then we compare our results with other published results. Okay. So that is the only way, but still it is not 100% we cannot get accurate results, okay? Because different system, different instrumentations, there are different type of instruments are used, different columns are used, different conditions in HPLC are followed in different labs. So most of them may not be identical. So to be identical, you need to develop the method. Okay, so for particular component, you need to develop and standardize a method which will help you in identification of the component. Any other questions? Okay, then we will move to the next session of the day. Okay, please stay with us. Content compliance by applying liquid chromatography HRMS e-replication strategies. He has optimized downstream process for large scale isolation of natural products and developed a methodology for fingerprinting of extracts with LC-UV-MS in natural products based drug discovery program. Dr. Abhijit's research group is investigating lichens grown on mangroves from the Gulf of Kutch and Gulf of Camber region of Gujarat to find bioactive molecules. Dr. Abhijit has 3 patents to his credit and more than 28 research publications from international journals. So with this short introduction, I invite Professor Abhijit to give us a talk. The floor is yours. Can you hear me all?
Yes, sir.
Good morning, everyone. At the onset, I would like to thank Srishti and this program for inviting me to this talk. And I would also like to thank Professor Anil Gupta for this presentation and all the team members of Srishti. Yes, so good morning. Today, I prefer also to have an interactive session so that if you have any questions, you can ask me in between also, or at the end also, whichever way you are comfortable with. I'm fine with both. I will share my slides. Let me know if you can see those slides properly. Today I will go through some of the understanding of natural product research and its contribution towards the medicine, and also like how analytical advancement have played an important role
in this aspect. That would be the key area of my research also, and I will show some highlights of how that analytical tools have improved the research and output. So we will discuss that. So I'm sharing my slides. So let me know if you can see the slide properly.
Yes, Dr.
Yeah, great. So let's get going. So as we discussed that we will talk about the analytical techniques in natural product research, and what we refer them as dereplication, and we see now how and why they are important. So just as a background, so that we will make a context properly, natural products and nature, to be specific, have helped human being from so many years. I think there is no second thought about it, right? And this is one of the milestone flow chart which I borrowed from one of the published literature here, that they have clearly highlighted a clear-cut milestone, the achievements of the natural product drug discovery program. I won't go back long, but you know, there were several Nobel Prizes that people have received, and the recent one was in 2015, where Professor Yutu received for the discovery of artemisinin and everything, right? And then recently in 2019, and say around that, China has approved the seaweed-based oligomanate compound for the Alzheimer drug, which is also suggesting that the people are still working on this area and trying to help in the medicinal field. You know, if I give you a brief summary, I would say in anti-cancer drug, 60% of molecules are from natural product. However, since 1990, the pharmaceutical company have started thinking about curtailing down natural product research. Curtailing down means they were not ready to invest more in natural product research. The reason, there are several reasons in my subsequent slides. I think we'll discuss them in detail. But the output of this is that nowadays the pipeline of these pharmaceutical companies are getting dried up, and you know, the new scaffolds are not available, new therapies are not available, new generation of the same scaffold is available. So that is an alarming situation, and I think that is why the natural product research and their advancement in natural product research is very important topic of research. I would highlight here. One more thing I wanted to show to all of you, if you are not sure about it, maybe some of you might be knowing this, that drug discovery and development is not a simple task. You know, it takes more than 12 to 15 years from hit identification to the molecule in the market. Huge investment also needed for this. If you can see my slide, yeah. If you can see the slide, you can clearly see on the right hand side this flow diagram that several compounds from various sources can be screened. We find out the biological target, we validate that, we optimize our lead, preclinical data studies that we needs to conduct on animal models, and then we have a good data in terms of minimum toxicity on animal models and sufficient efficacy. Then only it goes to the clinical trials, and which is nothing but the first human trials, phase 1, which looks after the safety part, which which on a small number of volunteers conducted on the healthy volunteers, which give a small amount of drug and see the hazard or see the safety perspective of that. In phase 2 trials conducted on the patient, small population, and see the efficacy and also the safety. In phase 3 of the large population, and the more as you are going from phase 1 to phase 3, the amount that you need in terms of money is exponentially increased. Then the FDA approves this particular data received from preclinical to clinical, all the data, and then they approve it for certain population, for certain treatment. And then what we see that, okay, US FDA and other EMP has approved so many drugs in this particular area. But that would have started maybe 10 to 20 years back, that process. Now, like here I mentioned, one new drug could cost maybe 1.5 billion US dollar. That was the available data based on that. It's a huge amount of money. And someone like the investor and all, if they want to put that money, they make sure that they get a good return. Definitely the blockbuster drug are getting good returns also, and that's how their discovery, their agency research and progress. However, the new companies are definitely, this is a big challenge, and are definitely will think twice before they invest in this particular area. Now, this is already a complex process, right? As we have already explained. Now, in that, if you talk about natural product-based drug discovery, which is again further complicated, because in drug design molecule, when you design a molecule for a particular target, you know the target already. But in nature is not producing any molecule for this particular target or medication. There are different roles of these molecules in nature, right? The secondary metabolite, what we talk about, small molecules also. So now when you don't know exact role, or when you don't know how this molecule is going to be used or our use, then it is difficult to connect these two things. This is a big task. Generally, people follow bioassay guided isolation. Bioassay guided isolation strategy is if we are looking for an cancer molecule, we check in the extract level, in the fraction level itself, that okay, this extract or fraction has that particular activity or not. If it possesses that activity, we go further in terms of isolation or isolated identity. It is a lengthy process. It takes more than 6 months, 1 year to find a compound which possesses that particular activity. And therefore, there are several bottlenecks or challenges, I would say, for natural product-based drugs. Now, the first challenge I am highlighting here is the isolation of known compound. What I mean by known compound is that, suppose in India, in our lab, let's say, we have isolated a compound from plant XYZ. Now, when we isolate the compound, we understand its structure, and when we check that if anybody has published this structure already or not, and the answer is yes, it is kind of a futile exercise. Unless until you have a very good, significant improvement in the biological activity. Otherwise, it is difficult to even publish that knowledge. So, that is one of the major challenges, and this is also the challenge faced by most of the scientists all over the world. That's one of the challenges. Second challenge is the large-scale isolation. There are several bioactive natural product molecules. They are in the shelf in the labs of different scientists, natural chemists, because they could isolate only in milligram level of those. They couldn't get in grams or kilograms level, because when you want to perform preclinical or clinical studies, you need a good amount of this molecule. good quantity. And if you want to market that, you need to have a sustainable production method for these molecules. That is also a big challenge. And the third, which I already briefed you about, is the biological target. Because these molecules are in the nature, and if you observe, there is most of the discoveries are the serendipitous discoveries. And that's the reason finding a target for a molecule is also a task, I would say, and it also takes a good amount of time. Now, there are challenges, and because of these challenges, the industry, pharmaceutical industry, are hesitating to put the money, invest their research in this direction. But the downside is that if no one invests in this particular research, then after a few years, there won't be any new scaffold, because there was a study conducted, and the scientists have found out that the imagination power of human beings is based on what they have seen at and that is only the small modification they can pursue or they can think based on their available knowledge, not drastic different from that. And so that is, I would say, is a big thing. And so someone should need to come forward and take up these challenges and solve it. That is the problem statement right now. The ideal would be academic labs and national institutes should play a major role here. And our also approach is the same thing, that we want to contribute in this area to our research. As I'm mentioning here in this slide clearly that on the top part is how this process is complicated, that from plant material or any other source, natural source, you extract, you fractionate, you find a compound, you get the compound, you isolate them, characterize them, kind of find out the activity, and then you come to know that, okay, after 6 months or 1 year or so, you isolated the same compound which has anti-cancer potential or anti-diabetic potential. Then you say that, okay, I rested almost 1 year without getting a new compound. So, when these things was there in the industry and academic going on, there parallelly there was an improvement in the analytical tools and analytical understanding. Hyphenation techniques were there, they evolved. Hyphenated technique means you are combining HPLC with mass spectrometer, that is called as LCMS. You are combining LC with NMR, that is called as LCNMR. And then several others also, GC, GCMS, CEMS, all other techniques are there where you are combining two things and then getting the information that independently those techniques couldn't give the information alone. That was something which was very important. That process scientists has used in natural product research. That is the term that is coined as de-replication. De-replication. We don't want to replicate the work that other person has already done by using different tools. So though the name is de-replication, but there are different approaches within that strategy, are there which can be followed. Basically, if I want to tell you what is it, it is a process of quick identification of known compounds, known chemotypes, which are already known. How we do that? So here, in one of the published papers, they have mentioned it as a bag of tricks. The tricks means you are trying to do some kind of quick analysis or quick magic and to say that, okay, this is there inside. It is not the magic by the way, it is just an analysis, a quick analysis. And that analysis has been carried out with these analytical tools, which are very important. So, if you see this diagram carefully, what we have natural product source. Suppose you have collected plants, 100 plants from different area of India. And now you want to see that which plant extract should have worked first. You need to prioritize it. 100 at a time you cannot work. Now how will you select? How will you decide? That is where these things play a role. There are bag of tricks which I said here, maybe n number of things, and the mostly used technique I would say is LCMS and the best. LCHR and the best. LCNMR also has been applied in several labs, but the cost is a little bit higher there. So there is also a trade there between the cost and the application. Now, if you observe, if you analyze, and that says that okay, no, this compound has been reported by some so and so person from this and this plant, then you don't work on that extract anymore, because most of these compounds are already reported by these scientists already. It's published. And they also reported that okay, this is the activity of those. Out of 100, let's say you have came across 5 plant extracts where you have not got any hit from the reported database. Then you say that okay, do you agree with me that your focus would be more towards those 5 things where no one has worked on, and the chances of getting novel things. So that's the reason that can be analyzed. Just make a fraction quickly, you extract it and make a fraction, or you can analyze the extract as it is. Done within 2 weeks. Depends on the facility what you have. So with a very good facility, you can do in the days. So rather than spending time like 6 months to 1 year, you can definitely spend 1 or 2 weeks to find out, should I work on this extract? That question is very important to ask. To save a lot of things, to save money, to save efforts, to save the field research field also. Because if you produce more new compound, I'm sure you will have more confidence in that research and all this. There is one statistical data, as you can see on the right hand side of the slide. The number of publications in different years from different countries. And as you can see that from 2004 to 2013, actually this review was published in 2014, and that was interesting review, but after that I didn't get the data related, but I'm sure the number of publications that has increased tremendously. As you can see that in 12, 13, number of publications and publications was like double than their previous years. And so you can see there is very well acceptance of this particular methodology in the scientific area. And that is what I think I wanted to highlight here, all of you, that how we are doing here in our labs and how that has helped us to, I would say, improve the process. Okay, this is a simple definition which I have brought it from the dictionary of the dereplication, which I have already explained it to you, but it is really clearly mentioned that it is a process of testing the samples of nature which are active in the screening process to recognize and eliminate them from consideration. Right? If they are already studied, if they are already studied by someone else. It is a stage subsequent to the preliminary screening in the process of discovery of pharmacological active substances in the mixture of natural. So by applying this process, by applying this strategy, you select the extract or plant specific for which you want to work further in detail, such a way that you will end up with most of the time. Right. So if I move forward, what are these different techniques and what are different methods? These are the few of them I have mentioned here. Earlier, it started with simple HPLC with in-house standards, where they used to record the retention time, UV profile, that they used to use for the identification. Then later on, when the LCMS came into the picture, they started using mass data. Mostly the accurate mass when it came, then the process has got a really good strength as the databases, worldwide databases, common databases can be used. And we will see in the next slide. There are a few other methods, like I have said, LCMS, LCMR, LCMR, and many more can be used. GCMS is also one of the techniques which can be used and which are being used for identification of this compound, for the volatile compound. Right? So, what are the different strategies, I would say? And what I mean by strategy, I just want to quickly give you a highlight of this. Like I said, it's the early identification of the molecules and the instruments which are like LCHRMS, LCNMR, LCMS, and all. Now, if it is MSMS or LCV, we generally use our own databases. We create these databases that are compounds, they are already isolated. Now, if you know the accurate mass, and I will have an example in our subsequent slide how to do that. I will show you. That data is being used, and you can search that throughout the different databases which are available online. Some of them are available freely, some of them are the comprehensive one. People have spent a lot of efforts to create those databases, so they are charging for that, use of that. You need a database, you have created some data, and then you are comparing it with something else. You need to search, right? And so that is what the database are important. Now, in a dictionary, if you find a or if you want to find out a word and it's a hard copy, then it is difficult for you to go to alphabetically and then find out that word, and then you get the meaning of it. But nowadays, in the Google era, you can simply search and then get the meaning of that word. So it is possible because you can search it. You know that search engines are very important. Same thing is here. If I can search that, okay, this molecular weight, this compound with a molecular weight, this and this, and from this and this plant, is anyone has reported any molecules earlier? Then you get the data, yes, 3 molecules from this molecular weight has been reported in this plant. Will it be used for your research? Definitely yes. And so that is how we want to use these kind of tools, and we want to speed up our research. We want to focus on something which is different, which is new, and which can contribute to this area, this research area. Yeah. Now, let's discuss about the strategy part quickly. So in this case, what we have fractionation of food extract, then we apply this de-definition analysis, and then we screen for bioactive. Then we get the active energy. So for example, here we have 100 plants, you de-duplicated, you selected 5, and then 5 you have conducted activity, and then you then perform the bioactivity. In another way, imagine that you have kind of change the second position of de-definition to the third position. Replace this. So now what will happen? All 100 you have to test the bioactivity. Then the bioactive ones you will kind of perform that de-definition, and then you go forward. What are pros and cons? What is so fussy about? Why we are so fussy? Because they give different information on it. That is why the strategies are. So strategy means what we are looking for and how we design our product. Suppose I am looking for an anti-cancer molecule, or from a particular, let's say, fungus. So I have 100 fungus in my lab. Screen their secondary metabolite. First of all, I need to know if there is anti-cancer potential. So what I'll do, if there are extract, I'll check that if these molecules are showing anti-cancer potential or not. And then I will replicate to see that what are those compounds? Are they known? And I'll ask, are they known for anti-cancer? And if the answer is no in any one of these questions, go ahead and isolate those compounds. While in the other case, if I apply dereplication, all the extract, whichever are the known one, I will eliminate that immediately. That might be anti-cancer molecule there also. So you are getting a different extract at the end. So these kind of strategies are very important and should be very thoroughly analyzed and put forward thoughtfully, because the end product will be different if you apply different. This is just one example I have shared with you. There are a number of possibilities that you can do with this kind of strategy. This is actually the point I wanted to make, because the diagram was very small. As you can see here, very well acceptance of this particular process in the next product research. And you can see the bar diagram very clearly that people have put in various strategies and various, I would say, purification processes during this years. These are some of the applications in this area. So it is not only related to the drug discovery. It is also, you can extrapolate to some other areas also. So for example, this is related to metabolites. This is again related to new compound by research. Then this is food application. They are using this strategy to find out the compound or metabolite which is present in this particular maple extract. This is also one of the applications. The tool is same. We're trying to find out the molecule. So here they have used this and they wanted to see how the lignans are there, flavonoids are there, the other tannins are there, and they analyzed that accordingly. This was also an important task in food application. Now, with this background, I have spent a good amount of time in background because I wanted to give you the importance of this particular tool and technique. And at the end of today's discussion, today's lecture talk, I want you to at least take this message at home that okay, there are new techniques are available and that can be used to find at the early stage nature of molecule, to identify the molecule and to give with a good amount of confidence in it through analytical tools and need not to isolate if they are known. You can have a different strategy to check it that. So, like I said, that the 3 different bottlenecks we discussed earlier, actually, if you can solve the first bottleneck up to a certain extent, that will be a good contribution to this area. And that is what we are doing. I have, in my introduction, that I have worked several years, like my PhD is in medicinal product research. So I worked on this particular coral when I was doing my PhD. And I have also worked on various plants in the Piramal and other organizations, and also microbes. Microbes are very important, and nowadays I have shifted. I'm still working on some of the plant, but I've shifted gears and I'm focused on the microbes. And I'll tell you why. Because there was a second bottleneck, that supply issue, the sustainable production. If I find out a novel compound in the coral, for example, and which we did, it was difficult to get that compound to the large scale production. Same applies to the plant. This I put it for the case of the Taxol also, it was very difficult. That's why it took so many years to develop that molecule. But the large scale production of that particular compound is very important. Microbes, in the case of microbes, if you have that microbe in your lab, might be fungi, it might be bacteria, Actinomycetes, and you can produce them in the lab, like fermentation. You may have a different type of fermentation, different volume of fermentation. And then you isolate those that you can have. Literally, you can produce a mass quantity. And I worked on one project which went to the IND level and performed the studies on the clinic. Just before the clinics, first in human trials, we have conducted all the studies. That molecule we have isolated up to 1 kg from maximum. So that's why I'm saying you that from the microbes we can do that, and that is why the distance are very important. Now these are some of the machines that I have used during my research, and you know they are really automated, and that's why you can speed up the process. You could do this job in 2-3 days because of these tools. Which here I'm showing you one of the liquid handler, which can in fact inject the sample of the column, and then you can collect it, the fractions, and you can simply drag, you can put it in an evaporator, which can dry it for you, and then you test the BIIS-11 and this is well bled through high screening for different type of acids such as anti-inflammatory, anti-cancer, anti-infective used to do separately microbial acid. But at least some target-based acids and some phytochemical acid that this phenotypic acids. Phenotypic acids can be done through this kind of technology, and then we can like speed up the process very nice. This is also one of the setup that we used to have. That looks a little bit shabby, I would say, the HPLC and the fraction collector, but that is how the labs are. You are running 24/7, and they are several connections, and then we are trying to collect all the information in one analysis. And so that is how we use. So we had with HPLC, and used to use a good amount of sample in that, and 90% used to go to collection. Fraction collector was connected to it through here. And you know that fraction collector we used to submit for the BIIS. And so we knew that, okay, at that time of, let's say in this case, if I'm not wrong, around 18-20 minutes, we have got a good bioactivity, and at that time what was the compound present, was the mass analysis, what the database talk about mass analysis. So for example, I'm seeing the anti-cancer activity against some particular cancer cell line. I found this active, and I found that okay, this compound is already moved for the anti-cancer acid. Did it? I could not work on that extract because I know that this particular extract has this compound which is showing this particular activity. Now we are all the dots connected, because the extract has activity, and most probably it might be this compound which is already reported for this. Now the same compound is present in the 10 extract, for example. Have that cluster analysis also grouping, and that can be done easily with that. One of the instruments is referred as HPLC, which is written beside advanced and HPLC, where we can have a good speed, and that is like ultra vibration chromatography that people are referring to, and that can be connected with the smaller columns with the lower particle size. And so this efficient separation is same kind of separation, or better separation in a small amount of time. So earlier, maybe 30-minute runtime here, we can reduce to 6-minute or 7-minute runtime in this case. Right? And so we can cluster the similar profile, and we can group them, or we can call it as a chemical fingerprint, which is present in the extract. And then we can club, and then we select one of them based on the yield. We had different detectors here, like ELS2, we had mass detector connected to the 1H2PLC. And so all the information about that particular sample we used to get immediately with the analysis. So that is how the things used to be carried out. If you want to speed up the thing, that you want to focus on the project which will end up with most of the time with the NCE or what we call as new chemical entity drug discovery. This is one of the projects referred as BIIS-11 that we work with Piramal. And there we have selected this for the large scale. After derivatization, we found some interesting masses and interesting compounds in this fraction. And so we went ahead for the isolation. And then we also performed some kind of enrichment strategy here, where we have kind of enriched the compound which we were interested in. That was considered as a novel based on the derivatization strategy. And so we enriched them in particular 40% enriched fractions where we could get the maximum compound in this. And then we further developed analytical and preparative HPLC method, where I would just want to highlight here the reason because some of you may not have exposed to the preparative HPLC. Generally, in HPLC we generally use the flow rate of 1 ml per minute. While in the preparative HPLC, we use 100 ml per minute. The column generally is 50 mm column, while our analytical column is mostly 4.6. So 4.6 mm column is like this small column, this width, while the preparative column is like a dumbbell-like shape. We have very good amount of weight there, and so that you can load a good amount of compound in that column, and you can collect a good amount of this. Because when you inject 100 ppm, that is 100 microgram per ml, and if you are injecting, let's say, maximum 100 microliter, injecting only 10 microgram in injection. That 10 microgram, if you collect the compound, you will take that microgram. So you need to inject more quantity so that you can get the good amount for the after purification. And so same thing here, we used to inject 1 gram of compound after enrichment. And that is how we used to collect a good amount in 1 or 2 injection itself to get a good amount of sample or compound in a pure. So after purification also there are several steps, and I wanted to quickly highlight those that we generally go with the purity check. When we check the purity, we check the profile also, UV profile, and then we see the fragmentation pattern. For in-house laboratory also we collect that, so that once you know the structure in-house, if you get the retention time, you get the mass information, you get the MSMS information, it is easy for the next sample to analyze it very quickly. Then the structural elucidation, which is also a very big area, I would say. And in analytical tools, structural elucidation, I would say, is the cream, a pinnacle. And that is the toughest as well as the interesting part of the research in the Supercritical area. Sorry for the typo here, that one should be superscript and 13C should be superscript here and not this. And however, these are some of the 2-dimensional NMR technique and 1-dimensional NMR, where we get information about the protons and carbon environment, and we assign the structure based on this data. It's a big area of research, and like I generally teach this for our master student here for a complete 1 semester. It's a big thing. It will be difficult to explain in one short this 1 hour talk. But just wanted to give you an idea that the proton and carbon NMR and this NMR technique has given a very good advantage, because because of this we don't destroy the sample. At the same time, we get the structure of this compound. That is very important. Unless you don't know the structure, it is difficult to proceed further in this branch. And these are some of the other things by IR we get in the functional group present, MS and molecular rate, molecule formula, purity estimation, HRMS with the molecular formula. I have an example for you as a case study. Yes, we will have time. I try to complete in the next 10 minutes quickly so that we can have a time for the structure discussion part if you have any question. So there are several projects that we conducted in this area and found out several NCEs. When we were at the earlier NCs per year, and we have also conducted the DBT sponsored project, mega project, where we have screened thousands of extract from different part of microbial extract from different part of India, and we arrived with selected several microbial extract that are deposited at NCCS. And now we actually got prepared a catalog of compound, and we have isolated some of the very potent molecules and selected NCEs, and that we have handed over to the further in the development for those molecules. So to give just a brief about it, that what were the different strategies we used. So like when I was doing my PhD, we used LC-MS strategy, where I used to create a library in-house by using the ion trap instrument, and where we have created a fragmentation of those particular molecule. And based on this strategy, they replicated several compounds, and we have isolated also several compounds through this in journal of natural product, which was one of the well journal in this area. Okay, so this was another case that I wanted to show, which is a recent one that we were working on one of the Indo-Sri Lanka collaborative project, which was funded by DST. And there our hypothesis was to find a novel compound from lichens present on the mangrove plant. These are the mangrove plant. This is one example, which is Avicennia marina, which is from Gulf of Khambat region. And there this is actually lichen, which is a different lichen, but we have also collected another thing. So this lichen is like a symbiotic organism between the fungi and algae kind of. No, they are symbiotic organism, and it's the complex thing. So it is not a symbiosis microorganism, but there are other microbes, they reside inside this lichen. And we wanted to explore endolichenic fungi. This is the fungi which is not forming this lichen. Because to grow this lichen in the lab is again a difficult task. We don't want it to work on something which with a large scale or sustainable production will be difficult. We wanted to work on something which we can grow in the lab in large scale. So we wanted to work on the endolichenic fungi which reside inside the lichen. We can produce this compound. In marine ecosystem or this kind of mangrove ecosystems, where there is a lot of competition for space, survival is difficult over there. And so these microbes produce a lot of chemicals. So chemical warfare is there. And that's why they are a good source of chemical. This is the reason that we have selected and chosen for our research. Sri Lanka is the best place to get the lichen because of that island and that environment. There were so many lichens. I visited Sri Lanka in this and I could find in the campus itself, there were so many lichens in the plant, on the trees, small plants also. And so it was the varieties of lichens that we collected and then published several papers from them. Now, with this, we got the endolichenic fungi, we fermented in the lab and prepared the extract. Here, the idea to look for the anti-cancer molecule, because most of these molecules are used in the microbial assays and they show the good antimicrobial activity, and generally the antimicrobial activities are turn out to be good anti-cancer activities. So, in both the direction we can pursue it in the large scale, and we could get a good NCs from this project also. One small case study I wanted to give you in detail for one thing, which is about HRMS, because you will realize you will understand what I'm talking about. It should not be only superficial talk. You should get some insights also. So, for example, this is just an example from textbook I have taken. So, there are 4 compounds with the mass 44. Now, if you can calculate, like if you take a 12 as a mass, atomic mass for carbon and 1 for hydrogen, so this total would be 44. Same thing for this. If you add a 16 for hydrogen, or 16 for oxygen, 4 for hydrogen, 20, and 24 for carbon, because 12 for each, so 24. So that's again 12. Same applies to this also, if you can calculate. So you see that if you find out with our mass type that okay, my compound has mass 44, there are so many molecules can be possible. You cannot de-replicate or you cannot identify like this. However, if you know the exact mass, and that is what is called as high, that we get through high resolution mass spectrometry. That is also referred to as accurate mass. And that can be up to 3 decimal places, 5 decimal places, based on your instrument resolution. In this case, let's take an example where we have got 44.029. Now we know the accurate mass of these individual atoms, elements. Now carbon has 12.00, hydrogen has 1.00784, and same for oxygen, nitrogen, sulfur, and all of that. If you add those accurately, you will get the accurate mass up to 3 to 5 decimal places. Now to compare the accurate mass with the observed mass. That is what it is done here for this molecule. And it was observed that this was the closest one with this. Observed. So now you can pick the closest one, the closest formula for our compound. That could be the compound of which is present in that particular. And that is how we kind of narrow it down the probabilities and we get a good success rate at the end for identifying this compound. I just wanted to give the power of this high resolution instrument that you can simply identify the molecule formula by doing this. And for this, you don't need to isolate that compound because we are separating it in a chromatographic HPLC column. And then we are sending one by one to mass. And that is exactly that's the beauty of this work, and you can get the best of both worlds. Their separation is there, and then you get the high resolution also in that case. So this is another example of the precise mass you can get, and you can assign the molecule for. By doing that, I would say this is really advancement of the analytical techniques have helped us to proceed further in this direction. We are working on several plants here, and we have worked on some of the projects and plants also. So that is also important because there are several studies that have been carried out from plant specimen, and we don't want the small labs in and around to work on the same thing again and again, and and isolate the same compound. So this is also one of the things that as an national institute we should work and we should collaborate and we should take the projects and quickly help other researchers to find the compound. So these 4 plants we have conducted the case studies and we have identified the already known compounds which are already present in that. While in one of the plants we observed, which is a Kachnar plant widely available here in Ahmedabad region, that we have collected. Paunia genus, Paunia variegata, Paunia parturia are the mostly common plant species. So I wanted to give you a case study. I know the time is limited already, 11:44 is there. So I'll quickly, 5 minutes, not more than that. I just wanted to give the both the things how we work, like based on the bioactivity guided isolation. At the same time, we wanted to apply the derivative strategy also. So this is generally we generate HPLC fingerprint, we conduct the dereplication strategy, we identify the compound quickly in the HPLC analysis itself to see that okay, this can be the probable structure or not. And if you don't find the hit, for most of the molecules. So for example, these were the hits that we observed with the database. We have procured dictionary of natural products also, which is not the hard copy, but the soft copy, where we can do the search, search based on the molecular weight, genes and species, based on the UV profile, based on the accurate mass, based on the activity, all sorts of criteria that we can feed and we can get the hit from this. We perform the fractionations like we generally do with the natural product isolation. We enrich the fractions which we wanted to focus on. We wanted to know that because we found some group of the compound which can be novel, which has not been reported from this plant earlier. And then we perform this particular analysis and we enrich them before the purification by refractive HPLC at 9 above. And then something that so many people are in waiting room, and I think that this is not allowing me to go to the next slide. Right. So we have finally we worked on that particular extract and we performed the preparative, semi-preparative we referred to because it was not that high scale, amount was less. And so we used a small column, which was 70 mm column. And then we purified the compound, we checked the activity and the structure illustration also where we got that formula. And then we are working on the NCE. We kind of know the scaffold structure, but we just wanted to, since we have isolated that the compound was very less and we could get only propanol and carbon and but we are working on the large scale isolation of that compound now. And it's almost there. This batch master should, I think we should get the structure. So with this, I will stop here. There are several report studies that I can share and we can discuss further also. But I think I just wanted to give you an idea, the overview of our research, what we are doing, and how these analytical tools have helped us to speed up the process in our research. In the acknowledgement slide, I definitely want to acknowledge my students, the PhD as well as master students. Without them, I couldn't have done some of these projects. And today I have explained you Ashwini's project related to that particular plant. And there are several PhD students which are there. They are working with me and the projects. I would like to thank my students and definitely my institute, Ahmedabad, for the funding and support. Department of Science and Technology from the particular grant of Like and Like from that. This is my group. Some of the students actually have left recently. They completed their masters, while PhDs are still with me, and some of the new students also joined. But not the recent one, but yes, the near recent photographs of my group. And thank you so much, all our audience, for listening me, for your patience. And I would be happy if you have any question. Thank you.
Thank you, Dr. Abhijit. The session is open for discussion. If you have any doubts, clarifications, you can ask. Students, hope you have all listened to the lecture. If you have any questions, please write to me. I will try to get your answers from Dr. Abhijit as soon as possible. Okay. Thank you. Thank you so much for joining us today. Tomorrow we have 2 more lectures. So please join us tomorrow without fail. Thank you. Thank you all.